I'd like to welcome you to Lecture 16, which is Chapter 12 on the central nervous system, Part 1. We're going to break this up into two parts, obviously, to make the information a little less bulky. So when we look at the central nervous system, we're now, unlike the peripheral nervous system, we're looking at just basically things that go on within the brain and spinal cord. And there's a feature of our central nervous system called cephalization. That means we tend to focus most of our primary control in the body in the brain and in the head region, and is not necessarily true for other creatures, but humans tend to be what we call heavily cephalic. Now, it's very important to understand the central nervous system is to look at its development, and this is to be very important understanding birth defects and other issues associated with the nervous system. So when we look at our nervous system, our total nervous system starts out as this dorsal area of skin that's focused heavily on the superior region of our body, okay, and this area called a neural plate in our skin, literally in the skin of the embryo, has this feature where it basically forms a dent in itself, and then we're going to see eventually grooves, and it forms this hollow tube in our body. So here's what the fetus looks like. You can see that's our little tail there, and this is our head region. Doesn't look like much. And this little area of skin-like material called your neural plate. And what happens, the neural plate is like a fold that eventually we're going to see moves into the developing embryo and eventually forms a tube. You'll see in the next picture, here's this thing called the neural groove, and these neural folds are going to kind of come up and together, and this is going to eventually form a solid ring that runs, you can see, throughout your back. There's your brain region. Notice how large it is right now, and this goes down to the tip of your spinal cord, which you can see right now. And as this develops further, look at your crest, because this is going to be your whole nervous system. You're going to, as a young embryo, where you're totally central nervous system, and eventually branches out into the peripheral nervous system. And here again, now you kind of look like the footprint of a weird alien, but no, this is your tail region, your head region, and there's now your neural tube. So this is basically going to be forming your early brain and your early spinal cord. Notice the hollowness. This is called a hollow tube. And this is where your cerebral spinal fluid is going to be and eventually form the ventricles of the brain. And you can see here, this tissue here, okay, will eventually be the area where you'll see your the spinal cord becomes protected by the tissues around it in your vertebral column. Now we can focus a little on the brain, the development of the brain, because again, this is very important to look at as our embryological brain develops. First, it starts out again, as a neural tube. It then swells at the end. So basically, your brain is a swollen, neural tube, is a swollen spinal column. And in some creatures, they have a couple of swellings, one towards the head and one towards the tail region. This is particularly true of reptiles and many of the dinosaurs. So we're going to see the brain starts as three regions called the prosencephalon, the mesencephalon, and the rhabdomencephalon, which we call in the adult the forebrain, midbrain, and hindbrain. And all of these are developed for a particular purpose. This is going to be your area of higher thought, interpretation, and things like emotion. The midbrain is going to be more of a region of processing. That means information that comes in and is then sent to the prosencephalon. Some creatures start at this level. This is a reflex area of where information goes, and the hindbrain is going to be very similar. This will be involved in very what we call a lower level, or what we call reptilian reflexes. So these two areas are primary reflex. This is going to be a much higher end center that we don't see on all living organisms. And then as the brain develops further, we're going to see now that we divide the brain further into what's called the telencephalon, which means the tail end, which actually in our brain is the front part, what we're going to call the frontal lobe. The diencephalon, which you can see is going to be two regions here. Okay, the mesencephalon, the mesencephalon and the myelencephalon. And this is because we start to see now myelin here, whereas in the brain, the surface can be covered with nerve cell bodies, what we call gray matter, rather than white matter, which is interpreted to be myelin. And don't worry too much about the functions of these in the ventricles. Just understand how our brain develops, and each of these stages of division can be responsible for very specific birth defects. And notice on the antelencephalon too, this is going to be the region where your vision is. And this is funny because when you look at shark and fish brains, their eyes are very big like this compared to the brain. You can see a lot of senses and neural capacity goes into their eyes rather than the thinking part of their brain, which is right there in the telencephalon. So here's a young brain. It's still not quite adult. But we can see now what the telencephalon has done. Okay, and there's your diencephalon there. These are both different degrees of higher order brain capacity. I'm going to leave your cerebellum out because that's more of your hindbrain region. It's like a little separate brain in itself. And then we have your midbrain. Okay, right there. And these take up most of what your central nervous system processing really is all about. This is showing a view of the inside of the brain, these ventricles, which started out as a little hole, okay, that ran throughout this neural tube. So you can see how very complex it got in the brain. And we have these regions called horns, because that's what they look like anatomically, that are filled with cerebral spinal fluid, which is produced in various areas. And again, this acts as like a cushioning in the brain and also separates the brain from the vasculature. It acts as its own cushioning and almost like its own little blood fluid in a way when you think about that. Now, when we look at the adult brain, and it's not colored like this, unfortunately, you have to kind of memorize this without the beautiful pictures, this is a brain this is your anterior region, your posterior view of the brain. So we can see here that the brain has these special features I'd like you to know. They're going to be called grooves and sulci, and that just means it's ridged because what the brain is, it's a layer of basically neurons with axons that go down into the brain, deep into the brain. They don't run this way. They run from the surface of the brain inward. Okay, and so we're going to see that these ridges here are actually like folds. So they're actually folds that increase the surface area of your brain. Because if you didn't have this, you'd have to have a skull basically the size of a small room in order to carry all that neuron capacity. So the brain is highly folded, and we can divide it into functional regions that we see here in the different colors that we call lobes and regions and areas, or else what we sometimes call gyri. Singular is gyrus. So you can see by the colors that different things go on here. You have what's called your somatic motor area. That means how do I take in and relate body movement to sometimes voluntary movement. Here is sensory information, baseline sensory. How did I associate that with, other body functions? So these are integration areas in a way versus this is just darn reflex areas. Okay, there's your visual cortex. Okay, and we have what's called the visual association, meaning how to relate vision to other things, just like these regions. And this is more of your reflexive visual center. So this kind of takes the reflex information of what you see, and this turns it into more of communication with the rest of the brain and with learning and other things. So these regions, there's your auditory region right there. And we have the association area and the reflexive area. That means incoming signals and how do I interpret those signals. Incoming signals, how do I interpret those signals and make use of them? So that's very typical of how our brain is segmented, very much like other creatures. So we look at the brains. We have these ridges or gyri, these shallow grooves, which are called sulci, these deep grooves, which are called fissures, and it just has degrees of folding. We then can divide the brain into 5 distinct lobes, which each lobe has various specified regions. Now, we're also going to see, again, pay attention to surface markings on the brain because this tells you where you are in the brain. And we're going to have a major central sulcus, which basically separates the frontal lobes from the parietal lobes. And this is very important because each of these lobes has a different function. We're going to have what's called a longitudinal fissure, which is sort of like a deeper sulcus that goes right into the brain, and it divides it into the left and right hemispheres, two halves. This is sometimes also called the lateralization region or part of the brain. So it divides the brain into left and right, or lateral sides. And then there's a transverse cerebral fissure, which separates the cerebrum. It's towards the posterior inferior part of the brain, so it separates the main part of your brain from what's called the cerebellum, which is like a mini brain that resides just above the neck. So here's what it looks like. There's your major frontal lobe in pink, and again, in a real brain, you won't see pink. Okay, there's your parietal, corresponding to parietal bone. Your occipital, which also kind of runs down here a little underneath the cerebellum, which is a separate, it's not part of the forebrain, it's its own little unique brain. That's right above what's called the medulla oblongata. There's your temporal lobe, and they cut inside the temporal lobe just so you can see the folding in there. And here's the grooves. This is where you find all the cells, not the axons. This region is axons. So the surface of your brain is literally the active, important component. And this is why the surface of the brain is very delicate. This is a superior view looking down on your brain. Here you can see the longitudinal fissure that divides each hemisphere of the brain into so you have a left frontal, left parietal, right parietal, your left cerebral, right cerebral hemisphere. So just, be aware of this, because you do function on two brains, and we do usually require two, but there are in some cases people are born with half and they somewhat function normally. This is looking at the lateral view of the brain. Again, look at your features here. You can see the dividing points. This is not always as evident, and you can pretty much see there's your cerebellum right there. So you can look at your different lobes. And again, this is not always evidence, and there is some variability in people, we know. But follow those major grooves to see where these regions are, and just use some general, sense to say, well, here's the occipital bone there, and that's the occipital lobe. So again, this is looking at the specialized regions. So anterior is what's called your motor complex. Remember the spinal cord? Okay, anterior vertebral column is motor, posterior vertebral column is sensory. So there's your sensory area, which goes right down the vertebral column. The vertebral column goes right up here anteriorly. Those signals go up there. So this is baseline information. That means just sensory information comes in here without interpretation. Motor information goes out here without interpretation. This is the interpretation sensory sections for each. And then, of course, we have emotion. That's around here, particularly interpretation of emotion, higher order thinking, memory. And then you have incoming auditory information, okay, and also processing of it. And then we have visual information, reflexive. That means I just see something and this interprets it, okay? And then we have areas of the brain that take all of this information and put it together in inner regions of the brains that we call nuclei and trunks that communicate information back and forth. Very complex. Brain anatomy is, you can spend years studying it and still not feel comfortable with it. This is looking at now the interior brain, so we cut it along a hemisphere. This region called the corpus callosum, you should be familiar with right here. It keeps in touch the left and the right brain, so they don't actually work separately, but sort of integrated together. You can see the ventricles right there. And this now shows some other regions where you have your primary olfactory or smell interpretation complex. So again, just look at this, appreciate the complexity, and just know the general regions of the brain. This is now looking at a coronal section, okay, or frontal section of the brain. and we're cutting right into the motor area. And this is kind of neat because you can see how different regions of the brain, from the superior to the lateral inferior, are responsible for different controls of the body. So you have your toes buried right in your brain, to your hands, and then your face, tongue, whatever, lips, eyes, all specialized. So when a person has a stroke, a physician could come in and see what actual damage is done and what that's going to be affected, or they can say, oh my gosh, vision is affected, so therefore this region might be involved. And the same is true for the sensory, which is more posterior. And again, it has very homologous regions, except there's a little digestive system control there for your visceral sensory input. Then looking at the brain from inside, a little more stuff is labeled. Again, just to pay attention to the medulla oblongata, okay, which is actually the connection between your spinal cord and the hindbrain. Okay, we have a connector right here with the cerebellum, and everything comes in. The pons is all a very important region because it takes all the information from here and from this body and basically filters it and distributes it through different regions and coordinates it with other input that comes in indirectly. That means not through the spinal cord. So again, a lot of complexity goes on here. So just look and appreciate the anatomy and its complexities. This again just showing you where areas like the hypothalamus is and things like that that you do hear about. Okay, and eventually, in other contexts. And a very important region of the brain you only see laterally, internally, is what's called the limbic region. And these are two terms to know: the thalamus, okay, and the hypothalamus, and the amygdala. These are all regions that associate incoming information with emotion. And this is particularly responsible for pain and pleasure, for sexual responses. This is the addiction region of your brain. That means if you're going to get addicted to something or adapted to something, this is it. And this is what makes humans in general very emotion-based creatures, particularly taste and smell come through these regions very strongly, and we associate it with, and we can learn to associate it with pleasure, pain, and even sex and other types of reflexes. So reflexes also get trained in here too, to whether a reflex is useful or not for us. I mean, this is why your reflex for peeing, the stretch receptors of the bladder stretch out, and you don't just pee in your pants because based on just your body wanting to, you can curb that in this limbic system area. So, what is the cerebellum? Because the cerebrum is the main part of the brain that we look at, that tells us all these different aspects that are controlled and integrated. But the cerebellum is like a mini brain, but it is about 10% of your brain. And this is an area that's somewhat misunderstood, and we're always learning more about. We do know that it's mostly under subconscious control, but it can be trained with, with experience. You can't really control it, but you can train it to develop muscle patterns. What we sometimes call muscle memory is really not muscle memory. It's basically how the cerebellum monitors what we do. And this is kind of funny because some people learn to walk normally, some walk with a limp. This is all based on that. Some are great coordination athletes, some know how to play a piano, some know how to fly a complex fighter jet, all based on this cerebellum. So the cerebellum, which basically means a little cerebrum, ellum means little or feminine, funny enough, and cere means a little, the brain. So we can see that there's various regions of it. We could also divide it into an anterior and posterior, which you don't have to worry about. But basically, it kind of messages from the spinal cord go there as they go here, and then messages go in and out here that all process with higher order thinking, but this is also responsible for reflexes with direct communication to the spinal cord. So again, it acts like a secondary brain, which is funny because we see this working as a separate brain and sometimes working with a little ganglion glump in the posterior part of the body in many reptilian creatures and fish. So that's the functional part of your central nervous system. No names, and basic functions. And please read your book on this, on generally what is the frontal lobe for? What are these things for? Please read that in your book. Now, when we come to the protection of the brain, it's probably the most protective structure in the body, the brain and the spinal cord. So your CNS is very heavily protected by bone, by what are called the meninges, membranes, by this watery nutrient cushioning called the cerebrospinal fluid, and by something called the blood-brain barrier, which we've talked a little earlier about when we looked at neuroglia cells or glial cells. So the meninges are this connective tissue membrane of varying thicknesses. Okay, I'm going to see that there's three major layers, and some of these layers can be subdivided, sort of like what we learned about with the skin and with the epidermis and dermis. So we're going to have what's called a dura mater, which actually means the tough mother, but don't take that wrong. The arachnoid mater, which means the covering or mother protector. Mater means mother or protector. Okay, that looks like a spider web. That's what arachnoid means. And then the pia mater, which means pia is delicate, the delicate layer or the delicate protector or mother. What do these look like? And this is a nice transverse section of the brain. You can see the hemispheres there, separate regions, left and right. There's the skin. There's the periosteum and the bone, the lower periosteum, and then below the lower periosteum, now you're in your dura mater, right here. That layer right there, which is directly below the periosteum. Now understand, tissue's attached to this area. But I mean, muscle and skin attached to that area. But once you get deep down here, this is pretty much a very loose connection. So you have your dura mater, which is basically sort of like an epineurium. It covers the whole, it's continuous connection around the whole central nervous system. And then below it is your arachnoid layer, and there's the spider web work of the arachnoid layer. And notice in the arachnoid layer, there's blood vessels, both veins and arteries, and around those blood vessels, they're not naked like that. They are covered with glial cells, okay, that basically totally cover those blood vessels, and anything that goes in and out of those blood vessels is controlled by those glial cells. And also within an arachnoid layer, you find, fluids. Again, the protective fluids. And then a thin layer on the brain, right there beneath the blood vessels, we find a pia mater. Okay, that innermost layer that directly makes contact with the brain and acts almost like an endoneurium. So, and actually it's really a perineurium that we find surrounding fascicles of nerve tissue. So what is cerebrospinal fluid in brief? It's a watery, usually clear kind of solution. Almost kind of looks, like it's a little glassy, a little hazy. Okay, it has less protein and electrolytes than blood plasma. It technically has a constant volume that's critically controlled by ependymal cells that absorb and release cerebrospinal fluid in various regions throughout the ventricles, the spinal cord. Okay, what it does, it allows the brain to float in this cushion so that when you come to a very fast stop, the brain comes to a slower stop because it has this really thick watery cushion, and it prevents it also from crashing into that inner region of the skull bones. Okay, it also protects from trauma because if you hit your head hit your head, the fluid takes some of the pressure off of that blow. It doesn't take it all out of it. And again, it's a nourishing area in which it has chemicals that have been selected by these glial cells, these oligodendrites, to cover the vessels, the blood vessels in this thing called the blood-brain barrier. This is looking at a sagittal view of the brain. There's your flow of your cerebrospinal fluid throughout this arachnoid pia mater layer. You can see where it all goes in the ventricles surrounding your spinal cord, within the spinal cord, and around. The problem with this fluid, if there's too little or too much, it can create problems. Too much can put pressure on the brain, and guys, the brain itself doesn't have sensory structures. Isn't that funny? It receives signals, but there are sensory nerves in the meninges that can feel pain. So when the meninges are swollen, you can feel devastating pain. And also when blood vessels are pressed on, you can feel pain too. So too much pressure could literally sometimes crush the brain or cut off the blood supply. Too little can make the brain vulnerable to damage, and also, a lack of that cushion, a lack of nutrients. Now, an important structure that we stress in this class is the choroid plexes. These are the regions that actually produce cerebral spinal fluid at a homeostatic rate. That means that it's usually monitoring the pressure of that fluid, and sometimes it doesn't do a good job on it. And remember earlier when we talked about nervous system and glial cells, the ependymal cells produce that in various clumps called choroid plexes. Okay, and don't worry about the ion pumps, but it uses a salt concentration gradient to basically pay attention to the fluid volume. Very precise system, but also it's subject to error. And then your blood-brain barrier, just a little more detail, and make sure that no neuron is in direct contact with blood-borne substances, which is a problem because sometimes you do want them to be. So what happens is the astrocytes and ependymal cells help to basically protect these neurons in there. They coat the blood vessels, and also they coat the neurons themselves to produce this barrier. So it's a very complex thing that makes sure that no certain nerve cell is in direct contact with the tissues or with the blood that leaks out of those tissues. So there's looking at your astrocytes. We also have oligodendrites that help to cover these and protect these neurons. So on that note, this was very brief, but please look at the functions of the lobes, the general functions, because this is important when we look at special senses too. Where's the information going? And what part of the brain handles it? And what do you look for with damage of the eye, of vision? And is it associated with the organ itself, the sensory organ, or is it due to the actual part of the brain that takes the baseline signal, the reflex signal, or processes that signal?